Welcome everyone. Hereby I open this academic ceremony which Evy Maas will defend the academic thesis, A Clash of Kings, Tools to Study Cross-Kingdom Interactions in the Human Gut Microbiota. May I invite you to present a summary of your thesis and the conclusions, please.
Thank you. Thank you all for being here. Thank you, my family, friends, and colleagues, and also people watching online. I will first give a short summary of my thesis, and I will do that in Dutch. So I will continue this talk in Dutch. So ik ga jullie even een samenvatting geven van mijn proefschrift genaamd Clash of Kings, Tools to Study Cross-Kingdom Interaction in the Human Gut Microbiota. Eerst een korte uitleg over de microbiota die gevonden wordt in het maag-darmkanaal. Deze zijn aanwezig in enorme aantallen, 10 tot de 14e, zelfs meer dan dat er cellen in het lichaam zijn. En deze hebben dan ook een invloed op de gezondheid van de mens. Bijvoorbeeld door de afbraak van voedingsstoffen die je binnenkrijgt via je voedsel. Ook werken ze samen met het immuunsysteem en bieden ze bescherming tegen eventuele ziekteverwekkers. De microbiota van het maag-darmkanaal bestaat uit verschillende kingdoms. Deze rijken zijn bacteriën, schimmels en virussen. En in onderzoek heeft vooral de focus altijd gelegen op de bacteriën. En daarom in mijn onderzoek heb ik ook gekeken naar schimmels en virussen. Antibiotica wordt gebruikt om bacteriële infecties te genezen. En dit heeft altijd heel veel succes gehad, maar heeft ook wat nadelen. Namelijk dat het ook de bacteriën verstoort die al aanwezig zijn in je lijf, dus ook de goede bacteriën. En dit heeft als nadeel dat dit ook een slechte invloed kan hebben op de gezondheid. Verder is er ook een toenemend probleem met antibiotica-resistentie. Steeds meer bacteriën worden resistent tegen antibiotica. Zo erg dat er voorspeld wordt dat er enorme aantallen doden veroorzaakt worden door deze resistentie, zoals je op dit figuur kan zien. En door deze toename in antibiotica-resistentie is er een vraag naar alternatieve therapieën, zodat die het gebruik van antibiotica kunnen verminderen. Antibiotica heeft naast de resistentie ook nog een ander nadeel. Het kan namelijk na gebruik van antibiotica kunnen schimmels vaak uitgroeien en deze kunnen een infectie veroorzaken en dit kan ook erg gevaarlijk zijn. In mijn onderzoek heb ik gekeken naar bacteriofagen. Dit kan een alternatieve therapie zijn voor antibiotica. Bacteriofagen zijn virussen die bacteriën kunnen doden. En op de figuur kun je zien hoe dat in zijn werk gaat. Ze binden zich aan de bacterie, waarna ze hun DNA injecteren in de cel. De DNA vermeerdert zich en daardoor worden nieuwe vagen gevormd. En uiteindelijk zorgen deze ervoor dat de cel doodgaat en openbreekt. En de vagen worden daardoor vrijgelaten en kunnen opnieuw andere bacteriën aanvallen. Het onderzoek naar bacteriofagen heeft lange tijd stilgestaan omdat antibiotica zo effectief waren tegen bacteriële infecties. Behalve in Oost-Europa, omdat daar door het ijzeren gordijn de antibiotica minder beschikbaar was. Dus daar is het onderzoek wel voortgezet. Maar daardoor is er weinig over bekend in de westerse wereld en zijn goede onderzoeken in het Engels zijn schaars. En een voordeel van de bacteriofaagtherapie is dat deze zeer specifiek is, waar antibiotica een hele groep bacteriën doodmaakt, gaan bacteriofagen enkel op één soort bacterie. Dus daardoor blijven de bacteriën die al aanwezig zijn in je lichaam die blijven in balans. Verder heb ik in mijn onderzoek gekeken naar de schimmels. Dat wordt ook wel de microbiota genoemd. En deze schimmels hebben dus altijd minder aandacht gekregen in het onderzoek. En dit is deels doordat het moeilijk was om deze te onderzoeken. Het is erg moeilijk om de schimmels te groeien in het lab. En met de opkomst van sequencing technieken is het dus makkelijker geworden om dit te onderzoeken en is er veel meer informatie beschikbaar. En daardoor kwam naar voren dat de schimmels, net als bacteriën, ook veel invloed kunnen hebben op de gezondheid. Nou, hoe ga je deze bacteriofagen en schimmels dan onderzoeken? Daar zijn tools voor nodig. In mijn thesis heb ik gebruik gemaakt van de TIM-systemen. Dit zijn in vitro modellen van het maag-darmsysteem. Zo kun je in het lab de maag en de dunne darm en de dikke darm simuleren. En hier zie je foto's. Links zie je het TIM-1-model en rechts het TIM-2-model. Deze modellen zijn computergestuurd en de pH en temperatuur worden geregeld. En er is geen zuurstof aanwezig bijvoorbeeld. En het doel van mijn onderzoek is geweest om het ontwikkelen van tools om de overleving en werkzaamheid van bacteriofagen in de menselijke darm te bestuderen. En om tools te optimaliseren om de schimmels en schimmelbacteriële interacties in de menselijke darm te bestuderen. Daarvoor hebben we eerst gekeken naar de overleving van fagen in TIM-1 en TIM-2, de in vitro modellen. En hierin zagen we dat de overleving voor verschillende soorten fagen anders is. Dat kun je ook zien in de grafiek. De rode en de blauwe lijn zijn twee verschillende soorten fagen. En we hebben gekeken naar een vaag die actief was tegen E. coli en eentje die actief was tegen Lacticacei bacillus. En hierin zagen we dat ongeveer 5 tot 10% van de fagen de dikke darm bereikt. En dus dat die overleeft in de dunne darm en de maag. Daarna hebben we gekeken in TIM-2, dus in het dikke darmmodel. En hier zagen we dat de fagen beïnvloed werden door de microbiota die al aanwezig is. En verder zagen we dat de bacteriën die aanwezig waren, dat die stabiel bleven na toevoeging van de fagen. Dus als je dit vergelijkt met antibiotica, waar de microbiota wel verstoord wordt, is dit dus een voordeel. Daarnaast hebben we gekeken naar de schimmels in het TIM-2-model. Hier hebben we de schimmels en de bacteriën vergeleken. En hierin zagen we dat de schimmels aanwezig waren in lagere aantallen en grotere diversiteit. Zoals je ook kan zien in deze figuur. Op links zie je de bacteriën en de spreiding daarvan is veel kleiner dan bij de schimmels op rechts. Verder hebben we ook gekeken welke soorten schimmels er gevonden werden. Dit was vergelijkbaar met andere onderzoeken naar schimmels in de darmen. Bijvoorbeeld Candida hadden we gevonden. En verder hebben we gekeken of verschillende soorten diëten een verschuiving kunnen veroorzaken in de schimmelpopulatie. En daarbij zagen we dat een hoog glucosedieet kan zorgen dat de soorten schimmels veranderen. Hiernaast hebben we ook gekeken naar schimmels die aanwezig zijn in verschillende ontlastingssamples. Hier zagen we opnieuw dat er heel veel diversiteit is, dus grote verschillen tussen alle verschillende proefpersonen. En de schimmels die we vonden zijn veelal afkomstig uit dieet en de omgeving. Bijvoorbeeld Saccharomyces, dat zit in brood, of Aspergillus, dat zit vaak in de lucht. En dit is een teken dat de schimmels meer op doorreis zijn in plaats van dat ze echt zich kunnen settelen in je lichaam. Hierna hebben we ook gekeken of de schimmels en de bacteriën interacties met elkaar hebben. Dus dat ze elkaar kunnen beïnvloeden. En we vonden in de ontlasting aanwijzingen voor deze interacties. En we vonden veelal positieve interacties. Maar omdat we enkel op één tijdsmoment hebben gekeken, is het lastig om er heel veel klinische relevantie aan te hangen. Dus daar is verder onderzoek voor nodig. Dit hebben we vervolgens ook gedaan in het TIM2-model. Omdat je hier de microbiota over langere tijd volgt, krijg je meer informatie. Verder hebben we de microbiota verstoord door het gebruik van antibiotica en fungiciden die schimmels doodmaken. En we zagen dat hierdoor de interacties veranderden. En dit laat zien dat als één van de twee verstoord wordt, dat dit ook een invloed heeft op de ander. De conclusies van mijn onderzoek zijn dat met behulp van het TIM1 en TIM2-model de survival van bacteriofagen in het maag-darmkanaal kan worden onderzocht. En TIM2 kan een goed hulpmiddel zijn om schimmels en schimmelbacterie-interacties te onderzoeken. En wat betekent dit voor de toekomst? Dat er meer onderzoek naar de werking van bacteriofagen nodig is om de vaagtherapie een serieus alternatief te maken voor antibiotica. En verder is er ook meer informatie nodig over hoe schimmels zich gedragen in het maag-darmstelsel. En de TIM-systemen zijn een goede tool om dit verder te onderzoeken. Dan wil ik graag het woord teruggeven.
Ja, hartelijk dank voor uw presentatie. And I will now switch back to English because we have some non-Dutch speaking members of the degree committee today. And before moving to the opposition, I would first like to introduce the supervisory team as well. They will not ask questions, otherwise you would be wondering who they are. So that's Professor Venema and Dr. Panders. So welcome. My name is Ralph Peters. I'll be acting as the pro-rector today to guide you through this ceremony. And then it's time to start with the opposition. The opposition will be opened by Professor Blaak. She also chair in humane biology, and she was chair of the assessment committee and also acting today as the secretary to the degree committee. Please, Professor Blaak.
Thank you, Mr. Pro-Rector. Dear candidate, at first I would like to congratulate you with this very nice thesis. It's very well written in a focused manner, so I read it with great pleasure. In particular because I also learned a lot on bacteria, viruses and fungi and realized how complex all the interactions can be. And also congratulations to your supervisory team. At first I would like to discuss with you the TIM2 model in the context of executing dietary interventions. Because it's a nice model which may resemble the human gut, and for instance it could be used as a kind of screening model, so what components may be promising also in human physiology. And you also execute some dietary intervention in chapter 4, and you have the experimental setup of TIM2 with in this setup an adaptation period, starvation period, intervention period. I was just wondering, during the intervention period you have then several conditions, and as a control condition you take the SIM medium. I was just wondering, to what extent does that really, is that really a good control, and does that represent also the conditions in the human colon?
Well, highly esteemed opponent, thank you for your kind words and your question. The SIM medium is the small intestine's efflux medium, so it's designed to simulate the efflux that comes from the small intestine. So it's already designed some years ago and optimized for the TIM2 model, so it does resemble the situation in real life quite well.
Yeah, no, I was just wondering, when I look at the composition, for instance, it also contains some proteins like casein. Is that not more digested in the small intestine then? Does it really present in that form in the efflux? Or?
I think to some extent it's digested, but also some part of it will end up also in the colon, I think.
Okay, yeah. So you do not need any pre-digestion anymore because...
Yeah, we don't do that with the seam medium, no.
Yeah,
But yeah, there are some examples of, I think, experiments where they do some kind of pre-digestion steps, but that's also usually more of the time if you add something to the seam medium, yeah, that needs pre-digestion to resemble the real situation.
Mhm.
Yeah.
And the C medium does not contain any lipids, or
No, I don't think so.
would that be a good option to add that, because
Yeah, it
possibly
can.
they may flow over?
Yeah, it
to the-
can be of an influence, but I think that will also complicate the experimental setup because, yeah, they will not be soluble. So maybe, yeah, this also complicates more the experiment than it makes up for, yeah, for the things you gain with that. And maybe it also depends on your research question if it's relevant,
Yeah.
if the fats have an influence. Yeah.
Okay, and then you have different experimental conditions, a low of a high carbohydrate, I think high protein, but also a high glucose, where you directly add the glucose to the medium. Is that a physiological condition?
No, not really. I think it was a bit of used as a more extreme condition because it's known that the fungi can use the simple sugar as a feeding source. So we would like to see if this extreme intervention has an influence so that later maybe you can use it when you do a more physiological relevant study.
Okay, yes, I agree, and I'm happy with that answer. When I look subsequently also at some of your results, also in figure 9, where you report the different fungi, you see that there's not that much effect of the high glucose condition on, for instance, Candida, and then you discuss it subsequently in your discussion that it may be controversial to several findings in literature where they use a high carb diet, or is that a valid comparison then?
Yeah, it is a different situation, I think. Also because here, you really only look at the direct effect of the glucose, so you take away a bit of all the other, substances. So it is a bit different, but still I would have expected that the Candida would like this high glucose diet, but it seems that other fungi liked it even more. And because these other fungi are not well described in literature, if people only looked at the Candida, for example, maybe they missed out the outgrowth of these other fungi.
Yeah, and when you have a high carbohydrate diet and you find correlations, of course, most of the glucose will be taken up in the small intestine also.
Yeah.
So does that suggest that other mechanisms may be involved in that relationships and not the
so
consumption of glucose?
the
per
high
se?
carbohydrate diet can contain different kinds of carbohydrates, so maybe not only glucose, so maybe another kind of feeding source is of relevance, but maybe also there is some interaction with the immune system and the host cells. So that makes it such a complex picture because there are bacteria, fungi and the host are all involving in this kind of situations.
Okay, am I allowed to ask a last question? Yeah. Going further to chapter 5, there you also compare the microbiota or microbiome, and you make a comparison between lean individuals and individuals with overweight, and you say there maybe not that much differences in contrast to literature. What I miss a little bit is the characteristics of individuals in the lean and the individuals with overweight. Can you tell something about that? Could there also be other factors responsible that you do not observe differences between groups?
Yeah, so there was not a lot of information known about these persons. So all the information that is known was written down in the thesis. So we only knew the age, gender, and the weight and the length.
What about the comparison between groups? Is it with respect to sex differences or gender differences? Is it comparable, those groups? Or
do you mean the groups are even divided, or
no, I mean
if there are
when
differences also between the sexes?
No, I mean just when you compare two groups, most of the time it's good also to present then the characteristics of both groups. I can imagine, for instance, that with a higher body mass index, that there are more females in that group. Or did you have a look at that?
No, so I didn't take into account the gender or the age for the different groups. Also because the groups are already quite unevenly divided. So this was the only difference we saw a little bit. But because it was not that striking, we did not really look further into this.
Yeah, but could it be that outcomes were less striking because there may have been differences in sex or age? Or?
Yeah, because the persons were not selected to be divided evenly across all the groups. So indeed, this could have had an influence. Yeah.
Okay, thank you. I'm happy with the answers and give the word back to the pro-rector.
Thank you, Professor Blaak. Then the opposition will be continued by Dr. van der Wijngaard. He's an assistant professor at the Tijdsgat Institute for Liver and Intestinal Research at the Amsterdam University Medical Center. And he was also a member of the assessment committee. Dr. van der Wijngaard.
Okay, thank you. Dear candidate, first I'd like to congratulate you. I read it with great pleasure. It was really nice to read. And I also enjoy very much that you put focus on, one of the focuses on fungi, which you can imagine probably, because I work on fungi myself. And when I write a paper or I discuss in a presentation, I always come to this point where I have to admit that only 0.1% is fungi, which is almost nothing. Mm-hmm. So what should I tell these people? Give me some ammunition to show that this is really relevant. Why is it relevant?
Well, thank you for your question, esteemed opponent. Well, I think in numbers maybe the fungi are small, but if you look at how big they are, they still take up a considerable space in the intestine, so therefore they can be relevant. And also because they are bigger, they are also more complex, so maybe they can in this way also have an influence on host health, for example.
Mm-hmm. Is there any examples already how they could be important in disease settings or in health?
Yeah, I think in, for example, IBD, the bowel diseases, there it is known that the fungi can be higher or lower, and this can cause some problems in the bowel. So I think that's one of the diseases that it's most studied in with respect to fungi.
And is that already beyond associations only? Because finding more and saying this is associated with disease?
Mm-hmm. Yeah, I think at least in mouse model there is some causality, yes, but in humans I think it's still quite new.
Yeah, I agree. Yeah, it's difficult. I completely agree. Yeah. Okay, but, I will use it. Thank you. So, I like the STIM model a lot, so I'm going to be asking some critical questions, but it's just to get the discussion going, because I wish we had it in our lab. It's really a nice model. But still there's some things I don't understand. So if we go to page 84 and 86, that's in chapter 4, then we look at the alpha and the beta diversity. And what you see happening very fast is the change in the beta diversity in especially fungi. Alpha also, in both in fungi and bacteria. And in fungi only, and not in bacteria, the beta diversity. And so I'm doubting whether it's not stable apparently, this composition. So how good is this model then if you cannot have a stable composition in 3 days? So why is it happening? And is it relevant? What do you think about this?
Yeah, I think it's happening because you take out the fecal samples from your body and you introduce them to this new environment. So I think it makes sense that it has to adapt to this new environment. And for the bacteria, it has been shown that it becomes quite stable. And also, all the characteristics are a bit pointed to this bacteria to make them stable. For example, the medium. And with the fungi, we saw, this was more difficult. So now we only did some first experiments to see how they behave. So we did not yet try to optimize, for example, the medium or some other characteristics, so that the fungi maybe also can become more stable. But I think it will always be a challenge because the numbers are much lower. So if one fungi grows a bit out more than compared to the other, the differences will be much bigger than when you look at the bacteria. So that indeed is a challenge. Yeah.
Now, one of my questions would be indeed, does it need some adaptation to look into the role of fungi? And that's what you actually say now. So what kind of
so, yeah.
What would you be thinking about then?
Yeah, maybe change the composition of the media, for example, but it's also a bit of a struggle because you also want to look at both the fungi and the bacteria, so you have to compromise a bit with
think
those
you're
two.
Right. Yeah, I agree. Now, if we stick to the same figure, figure 2, the alpha diversity, and fungi and bacteria, there's a huge spread. From what I get from the chapter is that you have 8 times ATIM inoculation, and that's the result we see. And the starting material is always this pool of 6 individuals, and it was mixed and aliquot, and then you take an aliquot and you start. So why is this spread so big in the alpha diversity? How can that be?
Yeah, I think there's still, you start at minus 16 hours with the same condition, but then you already have this adaptation period, so it seems that not all of the samples with the same, more or less same start condition move in the same direction. So the spread, I think you can see also at time 0, it becomes bigger. So already in that adaptation period, it seems that not all samples behave the same way. So I think something happens there.
And does that also say something about the way this first sample was taken or frozen, or I still don't get it. In my mind, it should be more close because it's just the same sample. And you do everything as good as possible, so I can see there's some diversity, but this is quite a lot.
Yeah.
So what about the sample then? Is it do you know how it was frozen, how it was aliquoted? Perhaps you did it yourself.
Yeah. So we take it and then we snap freeze it immediately in nitrogen, and then we move it to the minus 80 freezer until analysis.
And thinking back about my first question, where the composition changes so rapidly, did you ever check how well it is correlating to the initial sample?
We also took some, we analyzed also the fecal samples, but I don't remember exactly how that was, but I think you can also see some differences already from that,
Can I have one more question, or is time up? Okay. One last question, which I really am wondering about myself the most, so I'm really curious what you're going to answer. Figure 8 on page 143. It's about the short-chain fatty acids. I'm really interested now at this point on fungi and how much CO2 they produce during fermentation, because I think it's a relevant thing in IBS, because that gives pain to patients. And then I see your result, short-chain fatty as, of course, result of the same fermentation process. And what I see is that in the red bars is what is normal, the production of short-chain fatty acids, and then you add antibiotics, nothing changes. And then you add fungicides, and it goes down, which I think is great because I'm interested in fungi. But it kind of, if I make it very simplistic, antibiotics don't do anything, so short-chain fatty acids are only produced by fungi. I'm sure that's not true, but
Yeah, well, it suggests something like that, and I think there's really not a lot of information about this, so it was quite difficult to find also something in literature about this. But I think the fungi are overlooked when people look at this short chain fatty acid composition, and also the interaction with bacteria maybe. So I think it's quite complex. I don't think it's only the fungi, but it's the whole picture.
yeah,
Both
I
fungi,
completely
bacteria
agree. And I'm also
together.
still very happy with the result because it's challenging. It's intriguing. So thank you very much for your answers. Thank you.
Thank you, Dr. van der Wijngaard. The opposition will now be continued by Dr. Green. She holds a position as an associate researcher on phage therapy at the KU Leuven in Belgium. She was also a member of the assessment committee. Dr. Green.
Hello. So I'm going to change subjects a bit. First, I want to congratulate you on your hard work and this excellent dissertation and this presentation that I didn't understand but looked really great. So I'm going to go into a more broader question. So more research now is focused on the broader scope of the microbiome, like your own. We're no longer looking at bacteria. We are starting to look at fungi, viruses, the entire environment. It's an ecosystem. So now that you have a clear understanding of this complexity, do you really think it's possible to eliminate an opportunistic pathogen from our microbiome using phage, for instance? And what type of strategies would you use to do this, if you could?
Well, thank you for your question, esteemed opponent. I think it is possible, but indeed it is complex because when you test in the lab this kind of reaction, you put your bacteria and your phage and you eliminate all of the environment. And the TIM models already showed that the pH and the enzymes present can have a large influence, and in the real human body you also have an immune system, for example, that maybe even has an effect. But I think there are some quite success stories known where phage therapy could really help when no other things were working. For example, with the multi-resistant bacteria that were resistant against the multiple antibiotics. So I think it is possible. Also, I think I read about the fecal microbial transfer, where they remove the bacteria and only keep the phages. And when they add that to patients, they also saw, for example, a lowering of Clostridium infection, for example. So this, I think, also is some evidence that it indeed could work. And how would I do it? I think it's good to do good identification steps. So you need to know which kind of bacteria is causing the infection. So maybe by sequencing or something, that you really know exactly which kind of bacteria is causing some infection, and that you can really use the right phage for that, so that it really it's really suitable for that. So I think that will increase the efficiency of the therapy.
Yeah, so you you talked about FMT. So you said in these cases they only used phage and it was effective, or they used the rest of the microbiota and phage?
Yeah, I think they filter out the bacteria and other bigger particles,
Okay, so that tells you that it may be possible, at least against C. diff.
Yeah.
Right. Okay. You mentioned in your thesis that phage is commonly administered orally. I don't think that is the case. It's usually optimally administered at the site of infection. This, I think, is a less talked about but important limitation of phage therapy. So I think that how do you expect phage to actually reach the systemic system if it would be administered orally the same as antibiotics? Do you think that's an optimal way to administer phage? And will it get to the site of infection at the proper dosages if it is administered orally?
Yeah, I think that is indeed one of the struggles. So I think it will be able to transport across the gut epithelium cell into your system. But yeah, it is indeed a limitation, and that's also why this survival studies can be of importance to see what kind of doses is appropriate. And also I would suggest to do maybe subsequent experiments where you take into account also some cell work, some immune cells, for example, to see what happens there.
So you do think that that would be the best way to administer phage?
Yeah, it could also be intravenous, maybe that's more effective, but also if you look at practical, the practical way, then also I think orally ingested phages also should be investigated, because it's much easier for patients to take it in this way. So if you maybe protect the phage with a capsule or something, maybe it can be also effective in this way.
Yeah, I just think that, we tend to think of phage and antibiotics the same, and they're not. So I don't think they should be administered the same way. I don't think it would get to the proper site if they're administered orally. But I think they would be more, I agree, they would be more accepted if they could be just administered orally. One more question, if that's okay. So in your thesis too, you mentioned that the J1 phage, and in your presentation, I believe, was best in the stomach compared to the E. coli phage. So these phages tend to have specificity not only for the bacterial pathogen, but it seems like they also have specificity for the environment. This is another disadvantage of phage therapy, I think, too. So how do you think we can broaden the use of phage, knowing how specific it is for not only the pathogen, but the site of infection?
I think you would need to look into maybe a cocktail of phages, so that you can use different kinds of phages together, and in this way you try to broaden the spectrum. And also maybe these different kinds of phages, they act a bit different in these different environments, so that you have a higher chance of a successful match between the phage and the bacteria.
Okay, thank you. Thank you.
Thank you, Dr. Green. The opposition will now be continued by Professor Savelkoel. He was a chair in medical microbiology at this university, and he was also a member of the assessment committee. Professor Savelkoel.
Thank you, Mr. Prorector. Dear candidate, first of all, I want to start with congratulations for you and your promotion team, of course, for this very nice work. It's microbiology, so it's always good. On the other hand, I'm a medical microbiologist, so we use a lot of antibiotics. And now you challenged me in your presentation, because you said in the Western world we did not think of bacteriophages, and in East Europe they used them because they did not have the money for antibiotics, if I recall you correctly. Well, perhaps there are some specific reasons for that, which you didn't mention. So let's take step one. First of all, the question is, and I'll elaborate a little bit more on my previous opponent, is that you say that there's a cocktail needed when you want to do this as a therapeutic. But how many phages do we need to use them in real practice? Because it's strain specific, it's not species specific, and there are numerous different strains. So how does this cocktail look like?
Well, thank you for your question, highly esteemed opponent. Well, yeah, that's quite a challenge, I think. I think you will need some screening before an infection, and that you cannot make a bacteriophage like one fits all, such as antibiotics. So I think you will need to take another approach with this. So maybe there are maybe some strains that are more present in, for example, a hospital, like these antibiotic resistant strains that maybe keep popping up. So if you have these most common strains, then maybe you can find the corresponding phage for that one, and that you can make a cocktail of these most common strains. But I think that will differ maybe for the different hospitals, or maybe even more specifically. So I think indeed you need to take another approach for this.
Yeah, if I understand you correctly, you say in fact that for each infection we need to find out which phage is suitable to treat. But if a patient comes in hospital and he's ill and he has fever, and we don't have weeks to discover that. So I'm thinking then, how do we deal with that in real practice? Because if you have time, you need time to find the right phage, then the patient is either cured or is dead. So in both ways you are too late. How do you see that in practice?
Yeah, I think this sequencing and screening options will become much quicker in time, so that maybe can give you a bit more quick solution. But also maybe I think you can look into a combination of these antibiotics and bacteriophage together. So then maybe they can help each other. So you still are treating the patient immediately, and you also have the additional effect when the bacteria is antibiotic resistant, for example.
Actually, I was thinking you would use the TIM system now, or don't you think that there's a place for the TIM system in this approach?
Yeah, I think it can be used to design a more personalized medicine maybe. But if you take the time into account, maybe that will not be the best strategy. Then I would suggest to look more in the sequencing part to see then you have a much quicker result for identification.
And I imagine then that we have some kind of phage bank or so, and then we sequence the bacterium, and then we have an artificial intelligence module who will select for the phage. That will increase the shortens the time. But then, if I have time still, but then the next problem occurs, because in Europe it's not allowed.
Yeah, I know.
Because we have a registration of medicines, and that takes at least a few years. So let's assume this is possible technically, as we discussed, but do you have any solution for that?
Well, I think therefore this in vitro work is important. So in the TIM model you can also look at the safety, for example, and the pharmacokinetics, so you can really get more insights in how these phages act in this complex environment, and maybe that can help with the exception of these kind of medicines.
Yeah, but anyhow, you have. When do you know that your phage bank is completed? Because I'm a little bit afraid that it will continue and continue, and although they may be very specific, you may be always one step too late. Or do you think there's a solution for that?
Yeah, I think indeed with this AI or the sequencing techniques, they will become better and better. So in time, I think you will be ahead of time, but it's still a process. So I don't think at this very moment this is possible. But with the increasing antibiotic resistance, there is a need for alternatives. So maybe it's not always optimal, but you still need to look into these alternatives and try to develop these further so that in the future you can use them.
Yeah, because when we have an alternative, that will be great, of course, and you show that it is possible, and also from literature is known that there are quite some good examples from that, but they are very specific. And
yes.
This generality is, of course, the difficulty
Yes.
here. So I think we have a long way to go there, but we should definitely explore that. And then, how do you want to use it? Still, let's say you have this cocktail. Do you want to use it as a standard therapy or as a second option or only with antibiotic resistant strains, or have you any idea how we should use that?
Yeah, I think for now antibiotic maybe is still the first line of treatment. So maybe if that is not working, you can use it. But also I think the combination of the two I think can also be a good strategy.
But then you say first antibiotics and then second the phage therapy, which also gives you some time to find the right cocktail then. So that would be a new approach then.
Yeah, for example.
Yeah, that looks very, very, very promising. And if I understand correctly, we should eat more cheese because that benefits our bacterial microbiome. So for this moment, we don't have phages, but we can eat cheese. Or is that a strong conclusion for me?
I think it's always good to eat cheese.
Okay, perfect. Thank you. Well, I give the word back to the prorector.
Thank you, Professor Savikel, for bringing up the cheese. So the opposition will now be continued by Professor Jonkers. She holds a chair in intestinal health at this university. So, Professor Jonkers.
Thank you very much. Dear candidate, I also would like to congratulate you and your team with a very nice thesis. It was really very interesting to read. I think it's a very relevant topic. It's really important to pay more attention to the complexity of our microbiome, and you really contributed to that. Maybe I can first ask one of your panelists to read out the title of your thesis.
A Clash of Kings, Tools to Study Cross-Kingdom Interaction in the Human Gut Microbiota.
Thank you very much. I would like to discuss the title with you in two steps. First, the first part, A Clash of Kings. Can you motivate why you choose it, and especially the kings?
Well, thank you for your kind words and your question, highly esteemed opponent. Well, I choose it for one part was to get a bit of a catchy title to get the readers interesting, so that's also. But also, the kingdoms are clashing in your gut. So, the fungi and the bacteria and also the viruses are all living together. Maybe they are clashing together and fighting for the food that is coming in, for example, for the space, for the environment. So they are sort of clashing.
I understand that you're connected to the kingdom, and it's not a gender thing that you think that they are stronger there.
No, I mean the kingdoms.
Okay, thank you very much. I would like to discuss a little bit further with you this fungi. And I'm quite happy already to hear that you thought about how presentative these fecal samples and this preparation method, because it would be good maybe to optimize that a bit, because I noticed indeed that the composition of your microbiome in chapter 5 and 4 was quite different. So apart from this sample preparation, I wonder about the isolation method you used. Could it be, because this cell wall is quite firm, could it be that it was the isolation method is not good enough, that it beating, I think it's quite challenging to disrupt that. So was it really good enough? Are you quite sure about that?
Yeah, I think it was good enough because we got enough material in this method, but it was also a bit of a compromise because we wanted to look at both the fungi and the bacteria in the same study. So we quite chose this method so we could use the same method for both bacteria and fungi, because otherwise it would be really difficult to compare.
Or do you think that there are genera that you may miss, or?
Yeah, maybe in this way, maybe there are some genera with an even sturdier cell wall compared to others. That could be the case, But I think because the most important ones we found were also quite similar to what other researchers describe, so that also shows that maybe it is representative.
Yeah. Okay, but still I think it's very important that you make the first step to study the microbiota. And then you wanted really to study the interactions. You also pointed that out in your presentation. And you look to the positive correlation, and you have a nice table on page 116 showing some correlations there. Maybe the first question, because you described a few correlations from literature, you could not find those, but you find a few others. How can you explain the difference between what you find and what others find?
Well, I think this also takes back to the large difference that you find in individuals. So I think if we sample now these people here in the room, we will get maybe other results again. So it really depends on your diet and your environment. So if I would do it again, I would like to have more information about the diet and the environment of the test persons. And that can also maybe explain that these correlations are so different. But it also shows how complex it is.
I totally agree, because I was a bit disappointed. I expected way more correlations maybe. But maybe the most important thing is those are correlations. And in your title, you talk about interactions, interrelationships. Can you really conclude that you did study interactions? Or are they just correlations? How to interpret that?
I think from chapter 5, the correlations, because they are only taken from one time moment, it's difficult to say if they are real interactions. But in the TIM model, you follow them over time. So there you can see better what happens with these fungi and bacteria together.
And if you really want to know if there really is an interaction, how would you like to study that?
I think maybe you can also use the TIM model more as a screening maybe for these interactions. So you do these correlations, and then you take a specific correlation maybe in vitro to the lab to really see mechanistically what is happening, for example.
Okay, so we have to be a bit careful with the word interaction yet. We have to learn from that. No, I totally agree with you on that. And you already mentioned that there's quite some variation in the microbiota composition, and you mentioned in your presentation that it probably were passengers. But do you really think that they are passengers, or do we really also have to take them into account as colonizers? Because if they are just passengers, then I would not worry so much. What's your opinion?
I think you have both groups there. So I think a large group of them indeed is passengers, because it's known that some of the fungi we found are not able to grow at the characteristics that are found in the human body. So I don't think they will really stably colonize and be there. But that it is known also from maybe a study where they follow some people over, I think, 4 years, and the same fungi keep popping up over these periods. So then you could say this is kind of a stable community. But if your environment and diet maybe is the same during that period always, then still maybe they can originate from this diet, for example. So
can I ask? A last question. Okay, then I have a small burning question on the phages, because I was also quite intrigued by that, because I think it's very interesting to learn from that. And also you studied the survival and my neighbor already talked about that. And I was just curious, because you used the fecal microbiota of healthy subjects. What could we expect if you use the fecal microbiota of patients with a disease?
Yeah, I think then the microbiota is in this balance, I would say. And I think maybe because the phage is so specific, maybe the effect would not be so different unless the bacteria that the phage acts on is changed in this diseased person, for example.
I have the impression that I have to give the word back to the prorector. Thank you very much for your answers.
Thank you very much, Professor Jonkers. And the opposition will now be concluded by Dr. Camphora. And Dr. Camphora is an assistant professor at the Department of Human Biology at this university.
Thank you very much. Dear candidate, also for me, very kind congratulations. So very relevant and interesting thesis. I read it with really great interest, especially in relation to this, that you studied the I think still understudied kinks, so that was really nice. Also, congratulations to your supervisor team. I would like to start with a more general question on fungi. Yeah? So you stated quite strongly a couple of times, also in the intro, for example, that fungi have a big role in the host health, and you also said that, well, with the rise of sequence-based studies, it became clear that the fungal community can influence host health and is involved in several diseases. But then you continue to refer mainly on, as also one of the other opponents said, to more association studies, observational studies. So that does not say something about a cause of consequence. So I wonder, if you stated it strongly, what are really the mechanisms, how they interact with the human tissue, for example? Can you elaborate a little bit more on the mechanisms that you think are relevant for this?
Well, thank you for your kind words, esteemed opponent. Well, I did not really look into the immune function and host relation, but I can speculate a bit on that. I think you have some of the cell wall components of fungi. They can interact with the immune system, so maybe something can happen there. But also, I would say via the interaction with bacteria, also in this way they can influence host health. So not only to look at this fungi separately, but in relationship with these bacteria.
Do they produce also certain metabolites that might interact with?
Yeah, that's also one of the things. Yeah.
Okay, thank you. And then I would like to go to mainly on chapter 4. There you state, especially in table 1 on page 79, they'd shown nicely what kind of dietary interventions you used. And then I wonder why you chose for that. So high carbohydrate diet, high protein diet, glucose already mentioned, but also in one of the introductory sentence there was mentioned that, for example, high fat diet also influences quite a lot the fungal community, especially found in mice, at least found in mice. So can you elaborate a little bit more on that? Why you choose for those specific diets?
Yeah. So these experiments were meant also as a bit of a pilot experiment to see if the model was suitable for this kind of intervention and to look at the fungi. So therefore, we chose to adapt the standard feeding scheme with the high carbohydrate or high protein, so that to see what happens if you use these kind of standard adaptations to the diet. And indeed, if I would do future work, I would also look into these high fat or also some specific fatty acids that could maybe be of interest. But yeah, because this was preliminary work, we did not look into that yet. Yeah.
Okay, thank you, Ernst. And about the colonization of this fungi in the colon, for example, is there something known where they mainly colonize? Is that more the distal colonic part or the more proximal colonic part? And then also in relation to the model you chose for the proximal colon model. So can you also say something about that? What is the background why you choose for a proximal colonic model in that sense?
Yeah, for the exact place where they colonize, I am not sure, but yeah, I think most of the interesting fermentation takes place in the beginning of the colon. So I could think that for fungi this also would be the same as for bacteria.
Okay, thanks. And I'm looking at the time and I see that there's probably one more question. So I think that's a nice question to end up this defense. So I would like to ask one of you panelists to read out proposition 3.
Sequencing studies on the gut microbiota should include information on the microbiota in this thesis.
Okay, so it seems quite laborative to assess the microbiota. So would you recommend that also based on the findings of your thesis, and also that it's mainly known that especially in Western civilization, it's a really limited number of fungi present there? Would you recommend to do that really always, or would you say, well, we have to do that in certain subgroups, for example, in certain disease state or whatever? Yeah, can you say a little bit more on that?
Yeah, I think if you do it more, then also more information becomes available. So then you also know when it would be relevant.
Evy Maas, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. And I request that you and your company, the audience here present, will await the results of our deliberations and our return in this room. I adjourn.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Southside, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Hit the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future than we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. Southside, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
What do I do?
Nee, eerst daar en dan als... oh, sorry. English. En daar.
Let me reopen the session. Evy Maas, the degree committee here present has assessed the quality of your thesis and your defense. And in view of its positive verdict, and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Venema is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Please all rise.
First, Evy, do you promise to work in accordance with the principles of scientific integrity at all times and to be careful and honest, transparent, independent and responsible?
I promise.
Great, good. Then, by the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Evy Theresia Maria Maas, the degree of doctor, and grant you all rights attached by custom and law. And as evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Dan, beste Evy, Dr. Maas, wil ik graag een laudatio uitspreken. Als ik grimassen trek of het lijkt alsof ik knipoog geef, dan is dat omdat mijn contactlenzen vies zijn. Dus ik bedoel er verder niks mee, oké? Hier sta je dan, na iets meer dan 6 jaar nadat je bij ons begon. En dat je bij ons begonnen bent, dat was eigenlijk al raar, want we hadden die baan al aan iemand anders gegeven. Hè? Maar die liet me de dag erop weten dat ze ook een baan bij Danone gekregen had. En dat ze die baan prefereerde. Maar ondertussen had ik jou en andere kandidaten al gemaild dat we iemand anders gevonden hadden. Dus toen ik je hals over kop gebeld heb op je stageplek in Denemarken dat we je toch wilden spreken, was dat, dat was eigenlijk een telefoongesprek dat heel raar startte. Maar die mail die had je nog helemaal niet gezien. Maar goed, we hebben daar gesprek gehad en ja, ook jouw stage in Denemarken had dezelfde achtergrond. En uiteindelijk zijn wij ontzettend blij geweest dat je bij ons gekomen bent. Net als met alle andere AIO's en je 2 paranimfen kunnen dat beamen, heb ik jou in het diepe gegooid om te kijken uit welk hout je gesneden was. En of daar nog wat aan geschaafd moest worden. En of je kon zwemmen. Nou, en of je dat kon. Schaven was ook niet nodig. Met het projectidee ben jij aan de haal gegaan en heb je vrijwel zelfstandig jouw onderzoek helemaal uitgestippeld. En op praktisch gebied ben jij de AIO geweest die ik eigenlijk die het minst van mijn begeleiding nodig gehad heeft. Hoe anders was dat toen je het onderzoek op moest schrijven? Hè? Dat jij je proefschrift niet in iets meer dan 5 jaar afgerond hebt, is niet alleen maar te wijten aan corona. Het had vooral ook te maken met jouw moeite om dingen op papier te zetten. Of eigenlijk jouw perceptie daarvan. Want je bent namelijk redelijk perfectionistisch. En dat zat je met het schrijven, zat dat jou in de weg. Hè? Zozeer zelfs dat in de zomer van vorig jaar een moment was van ettelijke weken complete stilte. Ondanks mailtjes en appjes van onze kant. En heb ik via Rogier en zelfs HR geprobeerd aan jouw noodnummer te komen, omdat wij dachten dat er iets met jou gebeurd was. Dat noodnummer dat was echter afgesloten, dus dat hielp ook niet. Maar gelukkig was Monica, een van jouw paranimfen, zo slim dat je op WhatsApp kan zien wanneer iemand voor de laatste tijd online geweest is. En dat was nog diezelfde dag geweest. Nou, gelukkig hebben we dat allemaal opgelost. En hebben we jou ervan kunnen overtuigen dat de eerste draft van het artikel niet 100% perfect hoeft te zijn. Er zijn ondertussen 4 van de 5 hoofdstukken gepubliceerd, van de wetenschappelijke hoofdstukken. En ik heb er alle vertrouwen in dat ook dat 5e snel volgt. Ook heb je een nieuwe baan gevonden. sinds een paar weken aan de Universiteit van Wageningen als postdoc bij onze collega's van de afdeling microbiologie daar, waar je ook gaat werken aan een in vitro model, maar dan voor de microbiota in de dunne darm. Gefeliciteerd daarmee. That leaves me with thanking the promotion committee for their willingness and efforts to participate in evaluating the thesis and or coming up with questions for the interesting discussion that we had. I'd like to thank especially the two members from outside of the Maastricht University. René, thanks for the trip from Amsterdam all the way. It's good that nothing happened on the way here because I understand you don't have a mobile phone, so you would not have been able to reach us. Sabrina, also thank you very much. We've only met recently, but I've been following your work for quite a while. And when I learned that you are the mother of phage, I knew that we had to invite you for this committee. Thank you also for your trip from Leuven, which, although it is in a different country, is less than half the distance to Amsterdam. So with that, I give the floor back to the pro-rector.
Thank you, Professor Venema. Dear Dr. Maas, also on behalf of Maastricht University, I congratulate you with the degree you have acquired. And I would like to extend these congratulations to the supervisory team, but most of all also to the family, friends, and everyone present. That brings us to the end of the ceremony, so the official part is closed. And what will follow is a reception, and I would like to ask you to
the whole